My honor to introduce this next session, our keynote conversation with a CRISPR scientist. We have here today Dr. Feng Zhang. He's a core institute member of the Broad Institute at MIT and Harvard, an investigator at the McGovern Institute for Brain Research at MIT, and investigator at the Howard Hughes Medical Institute. He's the James and Patricia Poitras 63 Professor in Neuroscience at MIT and an Associate Professor at MIT. His team has trained thousands of researchers in the use of CRISPR genome editing technology through direct education, and by sharing more than 42,000 CRISPR components with 2,300 academic laboratories in 62 countries, all to help accelerate global research to benefit human health. So we welcome Dr. Feng Zhang, and he will be interviewed today by Carey Goldberg of WBUR. Carey covers health and science for WBUR, which is Boston's NPR news station, and she hosts the station's Common Health blog. She's also had a number of roles at the New York Times, the Los Angeles Times, and the Boston Globe, and she was a Knight Science Journalism Fellow at MIT. She has awards, including this year's American Association for Cancer Research June L. Beidler Prize for Cancer Journalism. So honored to have both on the stage and to have this interview coming up, this conversation. Just so you know, as with the last panel, you can ask some questions via our app. So we'll open that up. We'll also do some microphone questions as well for this session, so you can ask questions directly to Feng. So we'll do a little bit of both. So I'll turn it over to Carey.
Great. Thank you, Julie. Thank you. So Feng, welcome. This audience is somewhat glad you're here.
Well, welcome to Boston.
Yeah.
Sorry the weather is a little rainy, but welcome to Boston. Thank you for coming.
And so actually to jump off of that question, why are you here? I mean, I know you were invited, but personally, I'd prefer you spend as much time as possible in the lab. So what are you doing here?
Good question. It's an honor and a pleasure to be here, and I think I'm here to sort of talk about CRISPR. And it's really great to be here, given that we have such a diverse audience of scientists, non-scientists, biotech folks, and also possibly maybe some patient group here. I think as scientists, one of the questions that was just on the screen is what role should a scientist play in the public discourse of science and technology? I think one of the things that maybe I can do here today is to do some of the things that you think a scientist should be doing, to listen, and also maybe to help explain some of the things that might be helpful when we're talking about questions related to technology. So great to be here, and thank you for inviting me.
Sure. Yeah, it was interesting to me that people did not choose the function of reality checker for what the scientist's role should be, and actually that's what I'm thinking of as your role today. And so let's start with the big picture reality check. So people who haven't seen this Foreign Affairs journal outside, there's a big article by Bill Gates about basically saying gene editing will save the world, right? Like it will solve the world's biggest problems, from malaria to hunger with improved crops. It will make better cows, better chickens, better pigs, better everything, and so it should proceed full speed ahead. And so I wanted to hype check that with you. How do you see that big picture?
So I think maybe, I haven't read the articles, I don't really know what's in the article, but I think maybe one of the things that the article is really underscoring is how the recent advances in biology, both our understanding of fundamental biology, but also development of biotechnology, has come to a point where we can really use this information and the technology to do something that's good for the world. To solve some of these problems, whether it's disease or whether it's world hunger, and using not just in gene editing. Gene editing works in the context of many other technologies that work together to put forth these solutions. Is this new wave of biology, the present moment, that's really making it possible for us to start to think about how to tangibly solve some of these problems.
So focusing on the present moment in terms of the most important things that gene editing could do, so new therapies for diseases, improve agriculture, and others. What can we do right now, and what do you expect us to be able to do in the near future?
Well, CRISPR is a useful technology. So in the research realm, it's already making the work of many scientists a lot faster. We're now using CRISPR anywhere from the study of basic science all the way to developing therapeutic strategies and understanding disease mechanism, all the way to agriculture, building new crop species that may be drought resistant or insect resistant, and combining different traits of interest together. So CRISPR is making all of that possible. Now, some of the most tantalizing applications are how do we turn this into a useful therapeutic? How do we treat a disease like sickle cell or hemophilia? And that, I think, is something that a lot of scientists, a lot of really smart people are working on to make possible. There are still some challenges, but I think hopefully in the next few years, we're going to see the technology being tested in clinic, giving a successful result, and then being able to be developed more broadly for treating a lot of different diseases. In agriculture, I think this is also quite exciting. Before, it used to take a long time or a lot of iterative approaches to be able to get at a specific crop species. But with CRISPR, we can do a lot faster. Rather than spending many years to breed and cross different plants together, with CRISPR, you might be able to save several generations of that breeding so that you can get access to these important agricultural traits much, much faster. I think another emerging area where CRISPR is going to have a big impact is in the area of diagnostics. Using CRISPR, a different form, a lesser well-known version of a CRISPR enzyme called Cas13, it's possible to develop a rapid and also low-cost diagnostics for viruses, bacteria, and I think that will have a big impact on public health as well.
So that's what is true. I mean, these are all reality-based predictions. Let's talk a little bit about the splashy CRISPR stuff that's in the media and do a reality check on those. So for example, I feel kind of sheepish that I had a story on CRISPR and I put designer babies in the headline because I just wasn't confident that just telling a general audience that this tool is really revolutionizing biology in thousands of labs around the world was enough to get people to actually read the story. And so let's start with that. When you see media coverage that involves designer babies, what's your reaction? How do you feel?
Well, I think it's great that the public is taking a lot of interest in this technology. I've been hearing that friends of mine tell me that their grandmother called them to say that, have you heard of something called CRISPR? And my friends say, of course, I heard something called CRISPR. So I think it's really good that the public, there's so much interest from the public about this technology. And I think that underscored that the public really, there are a lot of important problems in the world, and the public is actively and eagerly looking for solutions that can help us address those diseases, world hunger, public health concerns, and so forth. So I think that's a really good thing. Now, specifically about designer babies, I think that's something that we're still pretty far away from that. With the current state, decades, with maybe even longer. With the current state of the technology, we cannot even treat a single genetic defect. To treat a sickle cell disease patient or hemophilia or cystic fibrosis, we're still a ways from that and still faced with many challenges. So to think about designing biology in such a way to give rise to higher intelligence or greater height or whatever trait that people may sort of fancy, I think it's pretty far away. Our understanding of biology is not even there yet. We don't even know how to make those changes. So I think the thing that we're really focused on, and I think a lot of people are focused on, is how do we solve the challenges associated with therapeutic development or application in agriculture? How do we make the efficiency higher? How do we make the immune system not sort of cause too much of a problem when we're delivering this as a therapeutic? And how do we make it specific so that we are correcting the disease-causing mutation and not other sequences in the body. And those are the things that we're focused on now.
So when the overwhelming majority of the audience cites eugenics as their greatest concern about CRISPR, what's your response to that?
I think these are all, none of these questions are not important. I think they're all good questions to raise, but the good thing is that we have a little bit of time before we really need to worry about it. So it's good that we're getting started. These are also hard questions. It's been around for a long time. With each wave of molecular biology technology, recombinant DNA, somatic nuclear transfer cloning, induced pluripotent stem cells, these questions come up again and again. And that just underscores how important these questions are, how challenging they are. We haven't been able to come up with a solution or an answer, but that just means that we need to think harder.
Okay, so let's do some science questions. I would encourage you to start thinking of your own questions and putting them up there. So CRISPR's been around since, what is it, like 6 years now, or so, 6 or 7 years. In that time, what has most surprised you about its trajectory?
I think it's really quite exciting to see how rapidly the technology has taken off. So the research community has been using this very broadly, really in all areas of biology, neuroscience, immunology, cancer biology, agriculture, insect biology, all sorts of areas. I think that's really exciting. But also, it's really interesting to see how quickly the public has gained excitement and interest in this technology. If anything, I think that probably surprised me the most is just how rapidly the public has sort of become excited. And that's a great thing for science, because we need that support, and also it motivates scientists to work even harder to realize some of these tantalizing applications.
Interesting. So I want to confess here that I asked Feng's lab members, what are the tough questions that you would ask him? So these come from your team. Is there likely to be a way to improve the paste function of Cas enzymes so that it becomes efficient enough to use therapeutically? Or will we probably end up using a two-part technology to introduce new sections of DNA, first cutting with a Cas enzyme, then using something as yet unknown to stitch in the new DNA?
That's a tough question.
Good. Good work, lab.
So CRISPR, CRISPR is a broad word, and it really refers to these little molecular machines that belong to bacteria. These are machines that are called Cas9 or Cas12 or Cas13. And for gene editing, what these molecular machines do is that they go and search for the mutation in the DNA. And then once it finds it, Cas9 will make a cut in the DNA. And it's that cut that triggers the cell to go and repair that cleaved location. If it's a mutation, it can allow you to give a new piece of synthetic DNA so that you can paste it into the genome and fix the genetic mutation. That process is inefficient. And in fact, that process really mostly works in cells that are actively replicating. And in cells that no longer replicate, like brain cells, the paste process is very, very inefficient. So that means new methods need to be developed. And how do we do that? There are a lot of different approaches that are being developed. For example, David Liu at Harvard and the Broad Institute have developed a system called base editing that allows scientists to change individual letters of DNA by performing a chemical reaction at that DNA letter. We've been working on something called RNA editing that allows us to modify RNA without depending on the cell's own machineries to make the repair. There are going to be new solutions beyond that. These are only ways to change individual letters. Sometimes you may want to fix a whole stretch of DNA sequence. The bacterial diversity is enormous. There are many, many more species of bacteria than we can even imagine. And so in order for them to evolve and to survive in diverse environments on Earth, in the stratosphere, in geothermal vents, in Yellowstone National Park, in all of the extremities, they have probably developed interesting molecular machines that remain to be discovered and harnessed to be able to manipulate DNA with much more agility. And so I think what we'll see in the future is that by exploring additional basic microbiological concepts or other biological diversity concepts, we'll be able to find new tools that nature has invented, and we may be able to co-opt them to make this pacing function even more efficient.
So you'll mine nature further.
Yeah, yeah. You can put on your Indiana Jones hat and
Yeah.
go treasure hunting in nature.
Go mucking around in bacteria. Yeah, great. So, and this could be a related question. If you could go back into the lab and devote all your time to one experiment, what would it be?
That would be really luxurious if I can just put all my time into a single project. I think I'll probably work on delivery. How do you get things, therapeutic molecules, into different parts of the body? Right now, even though when we talk about gene editing, we talk in broad strokes that you can use it to treat all sorts of diseases. In reality, the number of diseases that we can realistically tackle are much more limited. And that's because we cannot put Cas9 or Cas12 into the right cells efficiently. We can put things into the blood cells, so you can take blood out, modify cells, put it back in the patient. We can probably target the liver, the eye, maybe a few other small areas in the body. But the vast majority of organs, kidney, the brain, remain pretty difficult to get things into. And so developing new delivery systems, maybe ones that nature has invented and we can co-opt, or other methods using special chemicals that can penetrate cells will allow us to get access to those other tissues and realize the broad potential of the gene editing technology. So I think that's probably something I would spend time on.
Is your lab working on that?
We're doing some work in that area. Yeah.
And you've also recently been involved in a couple of companies, Pairwise and Beam. So what do they do, and why does what they do interest you?
So Pairwise is an agricultural biotech company, and the idea is to take advantage of some of the advances in gene editing so that we can make better agricultural products. How do we install better traits for drought resistance or insect resistance, improve yield? And those are some of the things that Pairwise is trying to be able to solve for the world. Beam, on the other hand, is a human therapeutics company. It's aimed at using these base editing technologies to either alter specific mutations on DNA or at the RNA level to be able to treat disease that we couldn't treat right now.
Got it. Okay. And let's get back to reality checking. I'm going to throw some things at you. You tell me if they worry you or not. Okay, so designer babies, very, very far away. What about Franken anything? Franken foods, Franken gene drives run amok, any implications that what's done with gene editing could leave our control?
I think we definitely need to be pretty thoughtful and careful about how we're implementing something that could have large ecological impact. Gene drive is a very exciting and powerful technology, but if we were to unleash gene drives into the wild, being able to think ahead of time, what are possible containment mechanisms? How do we reverse this gene drive are some of the things that we need to be careful about. So that's definitely something that I think we need to be very careful with.
Well, and work on that is underway, right? Like daisy drive. I mean, wouldn't the thinking be you don't release it until you know how to stop it? Yeah.
Yep. A lot of people are working on developing these containment solutions, which is very important.
And what about GMOs have gotten such a terrible reputation, and yet CRISPRed food seems much more benign, right? So could you clarify the difference and also what that could mean in terms of food?
Sure. So GMO usually refers to a plant that has a foreign gene incorporated into that plant. Whereas CRISPRizing a plant is simply accelerating the genetic changes that we can recombine into the same species that you would be able to get through conventional crossing and breeding, except we can do a lot faster using CRISPR. And so the end product of something that is CRISPRized is something that we can derive through natural means. It's just that we're making the process faster, taking fewer number of years as people would do traditionally. Whereas GMO, it really has a different gene in it. And those are some of the major technical differences between these technologies.
So could CRISPR make the old kind of GMO obsolete?
Hard to say, but I think in some ways they will probably, together I think they will address a broader array of different challenges. But with CRISPR, I think what will allow us to do is to understand the basic biology more, understand what can confer things that you may be able to do with GMO, but without having to make a GMO, by changing the natural sequences within these plants. And I think that's the long-term potential for the technology.
Okay, what about biohackers? There's been some buzz lately that people are futzing around with CRISPR on their own.
I think people need to be careful, and you shouldn't be injecting Cas9 into your body without thinking about what that will do to your body.
What would it do to your body?
So I do think that people need to modulate their excitement a little bit. Yeah.
But are you actually concerned?
I think I am concerned that if someone were to go find Cas9 and just inject it in their body, and that causes some undesirable consequence. So I think people they shouldn't do that. At the same time, it's hard to know who's actually going to go buy a bottle of Cas9 from somewhere.
Where do you buy it? Yeah.
And where do you buy that? And do that. I think what I'm most I think what's most important is that as we are thinking about developing therapeutic products, not to rush into trying this too early without sort of fully characterizing and understanding the potential of the technology. So when we're in the back room, we talk about gene therapy. And so I think one of the things that about gene therapy is that it took a long time for gene therapy to get developed, and there were some roadblocks and stumblings that have happened along the way. Right. And we can learn a lot from that process so that we don't repeat some of those same errors that we had done with gene therapy, with gene editing. And if we do that, we might be able to move actually faster than we think we're able to.
Right. Well, yeah, so the classic story is that gene therapy basically crashed after a patient death at Penn in like 1999 or
That's
something.
Right. Yeah.
So what can protect gene editing once it starts being used in humans from having a terrible outcome that could blacken the whole field again for a long time?
Yeah, so I think a lot of things can be done. We want to understand, first of all, how specific the technology is. And for anything that's being developed, setting a high reasonable standard for understanding the specificity of these things. Understand how the immune system will react to it. So this means not only testing on small animals, but also possibly in non-human primates, where the immune system is a lot closer to ours, to understand what the consequence of that would be. Understanding how we're manufacturing the reagents that we're going to be delivering to patients. Is it really pure? Are there other contaminants that are hard to detect but are maybe there? Toxins from bacteria, other modifications that may have immunogenic effect. So setting a high standard for quality controlling the reagents that are being produced that will be delivered into a human. And also understanding all of the technologies that surround the gene editing machineries themselves. When we're putting it into the patient, we may be using lipid nanoparticles to deliver into patient. What's the effect of those lipid nanoparticles, independent of the gene editing machinery, on the patient? How do viral vectors that we might use to deliver affect the patient? And understanding every single bit of the whole solution that we're turning into a therapeutic, I think is going to be very important.
And even with all that, there can be off-target effects, though, right?
Yeah, and I think off-target means that in addition to modifying the gene that you want to change, it also changes something else in the genome. So really thoroughly characterizing the system, how we design that CRISPR-Cas9 to go into the target gene in a variety of different cell types, I think is going to be important.
Yeah.
Yeah.
Great, so let me start. Julie, why don't we look at some questions from the audience? And let's do those for a few minutes, and then we'll have live mics going around.
Great, and I just want to flag, there was a question on biohacking, so we actually hid that already since you've just covered
Oh,
it.
okay, because I already covered it. Okay,
That was
so let's
appropriate.
do the most popular one by far, Feng, is what risks do you see from having, wait, is that it? What risks do you see from having a small group of people controlling the intellectual property for CRISPR tech? Huh.
Well, I think the major risk is that a small group would make the technology proprietary and prevent access from many different groups in the world. And so that's one thing that would be terrible to happen to the world. And so, in fact, that's the one thing that at MIT and the Broad Institute would really focus on, trying to make the technology available. We have been making the tools available to the broader research community, and also on a non-exclusive basis, making the technology available to all industrial partners, so that anyone who wants to use this technology to advance the development of science or development of products that would help improve the world will have access and be able to use it.
And so what will change after this patent fight is finally resolved? Well, anything?
I think hopefully, I think the ultimate outcome that will be good for the world is really this technology being able to move as quickly as possible. And then people can really broadly apply this to a lot of different applications, whether it's human health or agriculture or other biotechnology areas that people want to apply this on. I think making sure that the science and the development progress can keep going without being sort of slowed down because of this is quite important.
Great. Okay, next. Let's do, if gene editing is used to improve human health, will such treatment be able to affect countries with poor medical funding, or will it be financially restricted?
Right, I think that's one of the really important things that I think that actually I think about and discuss with people from time to time is equity of access. So in America, we have a very robust research system and also a very robust healthcare system, even though very expensive. There are a lot of experimental therapies and advanced therapies that can get developed. In countries where there is reduced resource, how do we make this technology be able to improve the lives of people there? And I think that's something that as we continue to develop the technology, as we sort of overcome some of the really expensive hurdles, like understanding safety, toxicity, all those things, the cost will come down. And manufacturing of these things as we scale up the cost will come down as well. So I think that those are some of the things that would help to make the access more available. In the area of diagnostics with Cas13, I think that's another area that we've been exploring very actively to see how do we get this diagnostic, which has the potential to be quick and also low cost, out to the developing countries where sometimes the needs are the strongest. And that's another thing that we're really working hard to make possible.
Great. So Sherlock that Jonathan and Omar spoke
That's
about.
right. Yeah.
Okay, I'm jumping to the second. Oh, good. It's the first question now. What keeps you up at night in terms of CRISPR? Excites and
to be
concerns
honest,
you?
probably my 1-year-old son
Yeah,
who wakes up from time to time. But mostly, I think I'm just really excited to be able to move things forward faster. There are a lot of hurdles that we have to solve. How do we make the system more robust? How do we deliver it? Those are the things that sometimes I stay up late and read about and try to think about new solutions so that we can solve the problem sooner.
And what about concerns?
I think we talk about designer babies and so forth, but I think we'll have some time, so I'm not losing too much sleep over that right now.
Until your kids are grown up. Good. Okay. How do you define what it means to do responsible research?
Well, I think doing responsible research means many things, and probably means different things for different people. But to me, I think as a scientist, we're in a very sort of lucky position to be able to do what we're excited and passionate about. But at the same time, I think it's important to be able to help communicate what we do with the public, help with education, help with sort of as people are thinking about the societal implications of technology, play a role in that. I think that's important. At the same time, I think it's also our responsibility to develop the technology and make sure that as we are solving some of these challenges with specificity, with toxicity, with immunogenicity, we are setting a high standard so that we're not sort of rushing to apply something, that we really think about it carefully and work with the regulators and everyone else around the whole sort of process to make sure we're taking careful steps. And then there are many other things. I think mentoring the next generation is important too. It's not just us doing research at this moment. We want the future generation to be excited about science and develop science and push technological solutions to solve real world problems. And so I think taking an active and also responsible role in mentoring the next generation is important for all scientists to do.
And that leads us actually to the young person question. How should young people get involved in shaping the future use or regulation of CRISPR?
I think many ways. The first way is probably to get involved in CRISPR research. We're always looking for young talent who are excited to learn biology and do research. Maybe they can start to do research and get excited about biology, not just CRISPR. There are going to be many new generations of technologies beyond CRISPR that will come along, and they can play an important role in both developing and shaping that technology. Those new technology, I think that's quite important, and that's probably a good place to start.
Yeah. Great. Oh, this is a fun one. Where do we draw the line between therapy and enhancement?
That's a very good question. I don't have the answer to that question. I think that's something that I think as a whole society will have to grapple with. The line it's not a black and white world. The line will probably shift depending on who we're talking with. I think that's an important question, and it's something that I think meetings like this in particular will help to foster the conversation.
Is there something, what would you hope to hear at a meeting like this, or from an audience, from the other speakers? And you also have contact with patients and other people who get in touch with you. What do you need to know from the public?
So I get emails almost every day from patients who heard about CRISPR or heard about gene editing therapy and want to get involved. And there are many different diseases. And I think one thing that has been quite inspiring and also humbling is to recognize that there are so many different forms of genetic mutations that what we have right now with Cas9 or Cas12 or even Cas13, we can only address a small slice of that pie. There are a lot of other changes that would be great to be able to make that will allow us to treat many more diseases. And so that's motivating. We're actively sort of on a day-to-day basis trying to develop new solutions to make those things possible. And then at the same time, I think sort of getting more people interested to work on contributing their own creativity and energy to solving these challenges is just as important. That's another thing that I work on.
So I feel like what I'm hearing from you is that even though designer babies and enhancement are fun to think about and talk about, we have so far to go just clinically that it's kind of moot, right? Like it's kind of like there's just too much to do just addressing disease.
Yeah,
right?
There's a lot of work to do just to be able to fix these single genetic changes that cause really devastating diseases for many families. I think those are the things that I think we have a chance of doing now. And those are things that we should focus our energy on.
And I wanted, speaking of those, not to forget malaria. How promising do you see CRISPR as an approach to malaria?
I think is very, so CRISPR is relevant in many different ways to malaria. A lot of malaria researchers are using CRISPR to understand the pathogenesis of malaria and also transmission of malaria. So that understanding of the basic biological process will unveil new drug targets that we can develop, not necessarily CRISPR-based therapeutics, but small molecules, protein-based therapeutics to be able to address. On the other hand, gene drive is a interesting and also exciting direction to take. It's an area that I think we need to be careful, but and there are certainly days I feel that it would be great to have no mosquitoes at all.
Yeah.
But if you really do wipe out mosquito population, how will that affect the ecological environment? Because you're removing such a large fraction of the biomass. That's something that I think we'll need to really be careful with.
So here's a good one. Should the products of CRISPR technology and GMOs be labeled differently? Sounds like you say yes.
I think so. I think honesty and being straightforward with what we're trying to produce is really important. So label, make sure everything is clearly communicated, and also at the same time providing enough information so that the public can interpret that information, understand it, and also be able to make their own sound judgment. I think is important.
Interesting. Yeah. So if anyone would like to ask live questions, now would be the time to raise your hand. We have people with mics up. I see one right here. Can come to you.
Given that the Chinese have already produced monkeys that have been CRISPR'd, and the first successful experiments on a human embryo that was supposedly of implantable quality happened last summer, why do you think that there's going to be such a long timeline before someone is actually going to pull the trigger on this? For the sake of, say, two desperate parents that really want a healthy child, and it's a wonderful story, and I'm just a little confused why you think the timeline is so long on that.
Well, first of all, the timeline is actually the timeline for treating some of these genetic diseases is actually not too far away from now. But that's not using CRISPR. So for parents who have a genetic predisposition for disease, there are other methodologies such as pre-implantation genetic diagnosis that can help them screen embryos to find ones that don't carry a genetic mutation. That's a technology that's available today, and folks are using that technology to be able to prevent certain genetic disease. So that's already there now. In the context of CRISPR, the reason that I think we should not be rushing into application is because we don't fully understand the off-target activity. So when we try to correct the disease-causing mutation in the embryo, there is the possibility that system may make some other changes in the genome. And those are the things that we want to avoid. It's possible that those changes will not do anything to the physiology or the development of the child, but we probably don't want to take that risk right now. And so as the technology gets further improved, we'll have better systems that are more precise, but also better analysis methods that can help us figure out, are any other changes introduced inadvertently into that genome? So until those new advances have been realized, I think that's why we should not be sort of taking this into editing embryos and treating disease.
But actually, just to follow up, that's certainly the should, but what about the can? I mean, so if I were a very careless parent who didn't care if my kid had off-target effects, would it in fact be technically feasible to already, or how soon would it be feasible to already edit a human embryo?
It is feasible. The technology is there now. Now, if a particular group rushes to do that, I would consider that to be irresponsible.
Great. Other questions in the audience? Raise your hand.
Relative to regulation, while local regulation is clearly doable, what about global regulation of this technology? How do we enforce global regulation?
That's a good, yeah.
Should there be some, yeah.
That's a good question. I'm not an expert in how we negotiate multilateral, multinational treaties to be able to enforce something. I think that's really hard. But I think what is encouraging is that the scientific community has quite high level of consensus. Just a couple of years ago, the very first international summit on gene editing was held in DC, and it convened scientists from around the world. And there was a remarkable level of consensus that research needs to move forward, but applications of this in areas such as therapeutic in the embryo should not be advancing yet. And so that's where we are. And I think that's really the best that we can count on for the moment being.
And there's another summit coming up this fall, I think, right? What
Yeah.
would you most hope to see at that?
I think continued discussion and also sort of the scientific community coming together to identify major challenges and then setting goals to be able to solve some of those challenges, I think are some of the good outcomes that can come out. And the focusing efforts to really do those things that are going to be productive and advance the possibility of actually treating diseases in the future.
Okay, I'll take another one from the poll. What's your opinion on DIY CRISPR kits that can be delivered to your house? You probably don't need them, and so you've never tried them.
Those are interesting kits, but those are, have very limited functionality. You follow a specific recipe, and it's hard to actually use that to do anything beyond what that specific recipe is. But I think there are also school kits for science classrooms, and I think those are good. They're helping students become acquainted and hopefully will excite them about wanting to study and do biology in the future.
Great. There was a question over here, people with the mics, but while you're on your way, let's ask this one. What is the greatest barrier for human application of CRISPR, and how can we overcome it?
Well, for me, I think one of the greatest barriers is delivery. How do we apply this into different organs to be able to treat diseases that affect those organs? I think that's a major hurdle, and I think a lot of research into different delivery approaches, viruses, exosomes, lipid nanoparticles, electroporation, other physical methods, those are things that will help move the field forward.
Great. Question?
Where do you believe the field is at in terms of having diagnostics to detect off-target effects in terms of the sensitivity right now, and how do we improve upon that?
Yeah, good question. So I think there are a lot of different detection technologies available now to measure off-target. And most of them probably work to a level of sensitivity of around 1 in maybe 100,000 cells. So if 1 in 100,000 cells have a particular mutation, we can pick it up. Of course, that sensitivity can get much greater, but the cost for making that analysis also increases proportionally as you get sort of deeper and deeper. We need new methodologies to be able to profile that off-target activity. And those are things that I think through more advanced biochemical methods we may be able to develop. And then at the same time, sort of doing these experiments in animal models and seeing that over a long period of time, how are there additional accumulation of off-target activity, and how do these off-target activity over the course of a long period of time affect the biology of these animal models will help us understand what's the biological consequence if an off-target activity is present.
Great. Here's a good one. What research is off limits today? Contrast the US and China.
I think I'm not too familiar with what the regulations are in China, but in the US, editing an embryo and implanting the embryo to give rise to a new baby, that's certainly off limits right now. Mm-hmm. Yeah.
Any other questions? Yep, over here.
Alternative gene editing technologies like zinc finger nucleases and talins? Do you see those as possible short-term options until CRISPR is fully tested in the clinic?
I think these are, so zinc finger, talins, CRISPR, these are all exciting technologies in the area of gene editing. They all work through a similar mechanism of gene editing, which is they find the DNA sequence and then they make a cut. And then once the cut is produced, it relies on the cell's own machineries to make the repair in the cell. All three technologies, I think, are quite promising. It will be up to the specific disease and then the specific molecule that's being developed to address that disease, and carefully assaying that and comparing the performance, specificity, and safety will be important to figure out which technology is most well-suited for that specific disease.
Great. One very popular question, can CRISPR be used for diseases related to the methylation state of DNA?
It's possible. So for example, there's a disease called Rett syndrome, which results from mutation in the gene called MECP2, and that gene binds to methylcytosines on DNA. CRISPR can potentially, in the future, correct that mutation so that we may be able to treat kids who suffer from Rett syndrome. There may be also ways to use CRISPR, not to cut DNA, but to modify methylation on DNA and to be able to alter disease state in a different way. These are directions that the researchers are developing very rapidly.
Interesting. Great, we have time for one more question. Is there anybody in the audience who has a burning one? Don't see anybody, so let's go to how do we balance the promises of CRISPR with the associated bioterrorism risks?
I think that's also a question that comes up in the media quite often. I think being responsible in conducting research is quite important, and also sort of figuring out what are the real concerns of CRISPR in bioterrorism is important. There are a lot of other bioterrorism approaches, and I think some of it may be directly applicable to what may be possible with CRISPR, but I think just doing a deeper dive to figure out what are the potential concerns and developing a solution for each one of them is how we should go about it.
Great. So please join me in thanking Feng.
Thank you.
Yes, we should have a podcast.
Thank you.